Hello and welcome to this video about polymers in nanomedicine. So the first thing you probably want to know is what is a polymer, because it's pretty important to this topic. So a dictionary definition is that a polymer is a chemical substance consisting of large molecules made from many smaller and simpler molecules. So to illustrate this, here you've got a selection of monomers. So on the left you've got individual monomers, just one molecule, could be ethanol, could be ethane, so just something quite simple. And these can then join together to form polymers. So here you've got 3 monomers forming a trimer, then you've got 5 monomers forming a pentamer, and 2 forming a dimer. And you can have any number, you could have thousands of monomers joining together, but collectively where you've got more than one monomer joined together, they're known as polymers. So how do polymers form? There are 2 main mechanisms. The first one is called chain growth polymerization, and in this, monomers can only be added to a reactive end group, so the length increases linearly. And in this, there are 3 main stages. Firstly, an initiation step creates this reactive end group, which can then go on to reactive monomers in a propagation step, where the polymer chain length increases each time the reactive end group is on the end, and then that adds to further monomers. And then when the reaction is finished, it can be terminated in a termination step, which basically makes the end group no longer reactive, so the polymer then stops being formed. The second type you need to know about is step growth polymerization, and in this, any molecule can react with another molecule. So instead of monomers being added to one reactive end group at the end of a polymer chains, monomers can instead react to form short chains, which then join together to form longer polymer chains. So as you can see here, you've got a few shorter chains forming. These chains can then join together to form longer polymer chains until you end up with a long polymer in the end. So, there is one special type of chain growth polymerization where there is no termination step. This is known as living polymerization, and in this, the polymerization will continue until all of a monomer is used. A different monomer can then be added to form a polymer with precisely defined lengths of each individual polymer chain, and this can lead to a formation of something called a block copolymer. And these are polymers formed of more than one monomer, with each block having a specific length, defined by basically how much monomer you added initially. And this is formed by living polymerization. So just an illustration here to kind of show you what I'm talking about. So you've got a block per polymer, so you've got a polymer chain of these 4 blue monomers here. Then when the blue monomer is used up, you can then introduce a red monomer, let's say. And then that reaction with a red monomer will continue until all the red monomer is used up. So then you can get blocks of precisely defined lengths of polymer chains. So that is a bit of background about polymer chemistry, which we're going to talk about in the rest of this video. And you're now probably wondering how this can be applied to nanomedicine. And one of the main ways in which this is done is through the formation of something called a polymeric micelle. And these are nanoparticles, and a nanoparticle is defined as between 1 nanometer and 100 nanometers in size, so that on the nanoscale. And these polymeric micelles are formed when block copolymers undergo self-assembly to form these micelles. And these micelles have hydrophobic heads and hydrophobic tails. So as you can see there, if we look back to the block copolymer we saw on the previous slide, you've got your hydrophobic tail in blue and your hydrophobic head in red. And these can then self-assemble to form micelles, as you can see here. So, before we talk about why and how these micelles I've just spoken about are used, I'm just going to spend a couple of minutes just to explain what nanomedicine is. Simply, nanomedicine is using nanomolecules, which are, as we just said, about 1 to 10 to 100 nanometers in size in medical applications. And they're quite widely used in cancer, and that's one of the examples and applications I'm going to come back to quite a bit in the next few minutes of this video. So, nanomedicine has advantages of improving the solubility of drugs, so they are more easily able to dissolve into your bloodstream and be carried to where they want to be. It can also make them last longer in the body, so they do not degrade quickly, so that means they'll stay at the site which you want the drug to be delivered to for longer. It can also release the drug at a controlled rate, so you don't get too much at once, and also can give more targeted delivery. So instead of having quite invasive techniques, so think about chemotherapy, for example, where one of the main side effects is targeting healthy cells. You can try and more target towards the unhealthy cells, so in terms of cancer tumour cells. So now we know a bit about what nanomedicine is, let's talk about how it can be used. So essentially what happens is a drug substance is encapsulated in the hydrophobic core of the micelles. And this helps to stabilise drugs, particularly if the drugs are hydrophobic, so won't normally dissolve or be as soluble in the bloodstream. It helps to stabilise them and solubilise them in the hydrophobic head of these micelles, so they're more easily able to be delivered to the target site. And when we get to where they want to be, they can either be released by physical stimuli, so if there's a change of pH, for example, or another way, which is particularly useful in cancer, is something called the enhanced permeability and retention effect. And this is basically the idea that tumour cells have larger pore sizes than that of healthy cells. So nanomolecules, which as you can see are between the 10 and 1000 nanometers of these larger pore sizes, would accumulate around these tumour cells and therefore target them more so than healthy cells. So this can be really quite powerful, these micelles, and particularly in cancer. So here's some examples. So they are small, and that small size can allow them to accumulate around tumour cells, as I just said, by the EPR effect. They also have a hydrophilic tail. This makes it more stable in the body, so it's able to be more soluble in the bloodstream than if you just injected a hydrophobic drug. And chemical properties can also be adjusted to modify a polymeric micelle. So it's now worth having a look at an actual example of the polymers used in these micelles and what they actually are formed of. So just to recap, they are formed of both the hydrophobic, so water-hating, and hydrophilic, water-loving section. So the hydrophobic, so head polymer, is generally PEG, something called polyethylene glycol, which is widely used. There are other used, but that's the main one, you see. And it's mainly used widely because it is versatile, because it's really, really hydrophilic, so will be very soluble in the blood. It's also biocompatible and biodegradable, so it is fairly safe, non-toxic, environmentally friendly, and it's also stable inside the body. And you can see the structure up here of PEG. So many different polymers can be used for the hydrophobic part of the micelle, and this generally depends on what best contains a drug substance. And some examples are given here on the right. So you've got like polypropylene oxide or polylactic acid. So there's lots of different types of polymers you can use for the hydrophobic part. So I now want to give quite a topical example of where nanomedicine can be used, and whilst this example has some quite complex chemistry involved and some structures which are a bit more complex than just the simple one hydrophilic part, one hydrophobic part to a micelle we've seen, it should hopefully still have some features and still be recognisable. The COVID vaccines is what this is going to be about, and these ones in use have lots of different technologies, but one of the more novel approaches which hasn't really been done before in vaccines is using mRNA. And this is both used in the Pfizer and Moderna vaccines, which are currently in use. And these both use this approach. And it basically works by carrying genetic code within the vaccine, which then tells cells in a body how to make certain proteins. And these proteins are then produced by cells in your body, and this can then trigger an immune response, hopefully helping us to gain immunity. So at the bottom right here, you can see the structure of the COVID-19 virus. And a main feature I want to emphasise and point out is the spike proteins on the outside of a virus, so the spike glycoprotein. And the mRNA vaccines essentially code to produce this spike protein, and then antibodies are created against it. So where do polymers come into this? So mRNA, when left on its own, just injected into your body, would degrade really quickly. So as you've seen with talking about cancer drugs, it's very similar in that actually the best way to make it more effective is to make it last longer inside your blood. So it is encapsulated in something called lipid nanoparticles. So on the bottom left here, you can see some structures of these lipid nanoparticles, and you can see that the RNA is encapsulated within quite a complex case, and there's a couple different types there, but you can basically see it's quite similar. You've got an inner and outer layer, and it's interesting to note that hydrophilic polymers such as PEG, which you've just seen previously, are located on the edge, and that's the green PEG lipid on the outside there. And here are the structures of the outer PEG lipids used in both the Pfizer and Moderna vaccine. And you can see it's quite similar to that bit there, simply the micelles we've been looking at. You've got quite a long hydrophobic chain towards the left, and on the right you've got the hydrophilic PEG chain, and that's essentially how these vaccines work. So I know that's only quite a brief overview, but hopefully that makes sense and just gives an example of how this nanomedicine is actually used in something which is hopefully everyone in this country is going to get at some point and make use of. So thank you very much for watching this video. I hope you enjoyed it, found it really interesting as well.